Hello, everyone, and welcome back for yet more human physiology. So, last time we talked a little bit about the relationship between a ligand and its receptor, but the thing we have not talked about is, well, what happens after the ligand binds to the receptor. So, let's go ahead and start discussing signal transduction here. So, what signal transduction is going to refer to is if you consider that the ligand, whatever that ligand may be, it could be a hormone, it could be a neurotransmitter, it could be something else. If that ligand contains a message, signal transduction deals with how the cell receives that message, interprets it, and translates it into a response. Now, the first thing for us to address before we really start getting hot and heavy into signal transduction mechanisms, what sorts of general things tend to happen when a ligand binds to a receptor? So there are several different ways that we can respond here. We will talk about several of these kind of as we go along, with our main focus being the first three here. But one thing that we can possibly do that we can go ahead and understand right now is that we said back in chapter 3 that ions like sodium and potassium and calcium, because of their electrical charge, they cannot pass straight through the membrane. They need a channel to let them across. But one of the things that I told you about ion channels is that those things are usually closed. So it's going to take a specific physiological stimulus to open an ion channel and allow a ion like sodium or potassium to move down their concentration gradient. Well, guess what kind of physiological stimulus would do that? Yeah, a ligand will do it. So if a hormone or a neurotransmitter binds to a receptor that looks like this one on the far left, what's called a receptor channel, then the binding of that ligand to the receptor will cause the channel to open up. And now suddenly sodium can enter the cell, potassium can leave the cell, calcium can come into the cell. So we can move ions down their concentration gradients in response to a ligand binding to a receptor. So that's just one thing that could happen. What's more likely to happen is that we are going to activate a type of receptor that is going to mediate a cascade of events downstream and inside the cell, and that will drastically change the function of that particular cell. So receptor enzymes and Gprotein coupled receptors definitely fall into this category here. The integrin receptor that we're looking at here on the far right, we are probably not going to get to discuss this semester. This is going to be, in some ways, how a cell responds to growth signals, and so basically the type of signal that would cause a cell to enter the cell cycle or a cell to kind of change its shape and start moving around. There won't be many occasions for us to discuss that, so we are going to just kind of focus on the first three here. So signal transduction and amplification is what our discussion is going to be about here. So anytime a ligand is carrying a message, there's probably a good reason for that, right? So for the sake of maintaining homeostasis, we are trying to get something across. We need to elicit a major change in order to maintain normal body function. So that message obviously needs to be strong. We need that message to be strong so that we can have a strong response, strong enough so that we can make the appropriate change. So the idea here, like we've said, is that the ligand is just a message. It is a message that needs to be read and translated so that we can give the appropriate response. We do not want to misinterpret that message, and even if we do correctly interpret that message, we need that message to be strong enough. We need that message to be amplified. So what you're seeing here on the far left, this little flowchart here, not necessarily a physiological example, but this is a real-world example of a signal transduction and amplification mechanism. So what you're seeing here is you are seeing a radio station broadcasting a signal, and then you are seeing a long wavelength radio picking up that signal and translating it into music for us to hear. So the external signal here, if anything is going to be the ligand, in this case, it's going to be the radio waves. So the radio waves are carrying a message that needs to be picked up. Just as we said that if a cell is going to respond to a ligand, that cell has to have a receptor for that ligand. So if our radio is going to pick up those radio waves, it has to have an antenna. So the antenna is going to be our receptor in this case. But the radio waves are not sound waves. So we understand that eventually we want to translate this into sound for us to hear, but that's not what the radio waves are. The ligand itself is not the eventual response that we're going to get. We need to take the ligand and turn it into a response. So that is what the transduction or transducer mechanism is about. So essentially the transducer in the radio is going to receive that ligand, receive those sound waves, interpret them, and transform them into something else. In this case, into sound waves that we can actually hear. But even after doing that, after transforming the signal into something that we can understand, the message is not going to be particularly strong. If you know anything about radio waves, especially AM radio waves, these radio stations do not broadcast at a particularly high wattage or power. So without an amplification mechanism, the message is going to be extremely quiet. We're not really going to be able to hear it. So that's why the radio also has a means of amplifying the signal after the transduction has occurred, so that instead of hearing a very weak signal, we hear a very strong signal and we actually have music for us to listen to. So any good cell signal transduction mechanism, we have to find a way to transduce the message of the ligand into something that the cell actually works with, and then we have to amplify it so that that response has the appropriate strength for what we need. So here's something that looks a little bit more physiological. So a new piece of terminology here that hopefully shouldn't be too troublesome to work with. The ligand is sometimes called the first messenger, so that should make sense. So that's why it shouldn't be too troublesome of a new term for us to work with. So the ligand, which is usually, as we discussed, usually going to be a molecule that cannot cross through the membrane, it's going to bind to its receptor or transducer on the surface of the membrane here. So we call it the first messenger because it's not going to be the only messenger. So part of any good signal transduction mechanism is going to be the conveying of the content of the first messenger's message to the other side of the membrane. So if you think of the membrane as being kind of like a wall, you bring a message to one side of the wall. Well, if you're going to get the message to the other side of the wall, you've got to have kind of an intermediary there to convey that message from one side to the other. So the transducer here, the receptor, is going to transduce this by producing on the other side of the membrane a type of messenger called a second messenger. So this is also going to represent how we get our amplification, because the transducer, which you can't really see it in this picture, but the receptor / transducer is usually linked to a amplifier enzyme. So that enzyme is going to rapidly produce a very great number of second messenger molecules on the other side of the membrane, the intracellular side. So by doing it this way, by having the first messenger bind to the receptor there, the transducer activates an amplifier enzyme on the other side. The amplifier enzyme rapidly produces many second messenger molecules. Those second messengers are going to have effects on proteins inside the cell. That's what you see here, these little blue squares here. These are target proteins that when they interact with those second messengers, those proteins are going to in some way change their function, and it's that change in function which is going to produce our desired physiological response. So this picture is a little bit better here in terms of appreciating how those second messengers get made. So here you can actually see how the transducer or receptor, after it binds to the ligand or first messenger, that receptor is somehow functionally connected to an amplifier enzyme which will rapidly produce those second messengers. And you can see over time how many different types of second messengers get produced, all from one ligand molecule. So one ligand can produce hundreds, thousands, if not millions of individual second messenger molecules. So if, say, one ligand produces 1,000 second messengers, then we have amplified the signal 1,000-fold. If we did not have this amplification mechanism and say we needed 1,000 second messenger molecules in order to kind of hit our threshold for a response, if we did not have that amplification mechanism, we would need 1,000 ligand molecules to give us 1,000 second messengers. But because of the second messenger amplification system here, we can get a very strong effect even with a very small amount of ligand. And this is going to be important in chapter 17 because we're going to see how a lot of our endocrine glands, like the pancreas or the thyroid, generally do not produce a very large concentration of hormones. So we can get very strong effects even though we're not using a whole lot of messenger. So we want to look at two specific signal transduction mechanisms here. One of them we're going to look at a lot more than the other. So let's start with the one that we're not really going to see very often this semester, but it's still fairly important. So this type of mechanism here is what is called a tyrosine kinase receptor. So this looks very similar to the amplifier, kind of generic amplifier enzyme example that we looked at on the previous slide. So a tyrosine kinase receptor has two different parts to it. It has the receptor part, which is facing towards the extracellular fluid, so that it can interact with a ligand like, say, insulin. And it's worth mentioning that the insulin receptor is a tyrosine kinase receptor. And then the receptor part is hooked up to the amplifier enzyme, in this case, what is called the tyrosine kinase. So what happens here is that we don't really have a second messenger per se, but rather what that amplifier enzyme is doing is modifying the protein directly. So we don't really need the second messenger so much in that regard. So the tyrosine kinase, a kinase is an enzyme that adds a phosphate onto a protein, and a lot of proteins can be activated by the addition of a phosphate, or a protein can be inactivated by the addition of a phosphate. So we have a lot of control over the function of intracellular proteins when we activate a tyrosine kinase receptor like this. And like I was saying, insulin and the insulin receptor represent a mechanism that looks just like this. So this is going to represent how we get glucose into our cells. And we will talk a lot more about that in chapter 17. So the other major signal transduction mechanism, the one that we are going to see most commonly, is called a G-protein coupled receptor mechanism. So this one is going to look a little bit complicated, so I don't want you to freak out about that just yet. We're not going to be responsible for understanding every single little detail here, but there are going to be some things that I do want you to take note of. First, look at this picture. So find the cell membrane. The cell membrane is here in dark purple. The G-protein coupled receptor is this thing that you see passing through the membrane 7 different times. So if you follow my arrowhead here, 1, 2, 3, 4, 5, 6, and 7. So G-protein coupled receptors tend to pass through the membrane 7 times. So that is just the receptor. That is the G-protein coupled receptor, and you can see how a ligand could bind right here. So attached to the G-protein coupled receptor is, hence the name, what is called a G-protein. A G-protein is a complex of 3 different proteins. Those proteins are called alpha, beta, and gamma. So when the ligand binds to the G-protein coupled receptor, we are going to activate this complex of G proteins, and it is going to slide over through the membrane and activate an amplifier enzyme. In this case, the amplifier enzyme is called adenylyl cyclase, or you sometimes might called adenylate cyclase. So adenylyl cyclase is going to utilize ATP to produce a specific type of second messenger called cyclic AMP. So it looks like camp here, but it's called cyclic AMP. So cyclic AMP is just about the most common second messenger that exists. So cyclic AMP is going to act as an activator for a type of kinase enzyme called protein kinase A. And similar to what we saw on the previous slide, when protein kinase A is activated, it can phosphorylate a number of different proteins inside the cell, and that will in some way change our cell function and give us our cell response. So G-protein coupled receptor pathways tend to do things like activate these types of second messenger pathways so that we can synthesize a new protein, get a muscle contracted, open an ion channel. So there's a lot of different things that we can do with G-protein coupled receptors, and we will be sure to mention those examples as we come across them in the future. So what you're seeing right here is just an alternative form that a G-protein coupled receptor might take. So not every GPCR pathway is going to look exactly like what we saw on the previous slide. So this one's just a little bit different. So again, you can see how our G-protein coupled receptor passes through the membrane 7 times, and it is still hooked up to that 3-protein complex G-protein here. Except this time, our amplifier enzyme is not called adenylyl cyclase. This time it is called phospholipase C, PLC phospholipase C. What it is going to do is it is going to take one of those phospholipids in the membrane, and it is going to convert it into 2 different molecules. The first called DAG, or diacylglycerol, which can go and activate another amplifier enzyme called protein kinase C, and it will do a lot of the same sort of things that protein kinase A did. The other second messenger here is called IP3, or inositol trisphosphate. So IP3 here can go to the endoplasmic reticulum and release a lot of its stored up calcium, and calcium there can act as a second messenger itself. If you'll recall back in Chapter 3, we briefly mentioned how the endoplasmic reticulum, in addition to its functions in synthesizing proteins and lipids, it also acts as a big reservoir for calcium. So now you can kind of see a little bit of the payoff for that. So if calcium is released from the endoplasmic reticulum, that calcium can start doing a lot of things inside the cell, and of particular importance, this is going to be very similar to what we see when we start talking about muscle contraction. Generally speaking, a muscle will contract when there becomes a lot of calcium available in the intracellular fluid, as you see here. So one of the most common signal transduction mechanisms that we are definitely going to see when we start talking about the nervous system is when we open an ion channel. We mentioned previously how this may be something that we do when a ligand binds to a receptor. So these types of ion channels that open in response to a ligand binding to a receptor are called ligand-gated ion channels. They are gated ion channels in the sense that they are closed most of the time, and we open them or ungate them when we provide the appropriate stimulus. And because this is called a ligand-gated ion channel, it tells us that the stimulus is a ligand binding to a receptor. So this can happen in several different ways. Either the ion channel itself is a receptor, which is what you see here on the far left. The ligand binds to the channel and the channel opens, so that should happen very quickly there. Or alternatively, we can open an ion channel through a G-protein coupled receptor pathway, which is going to be a little bit slower. So as we're going to discuss later in the course when we start talking about the nervous system, opening an ion channel is not a trivial thing. It's going to have very serious implications for how a cell behaves. Because when you open an ion channel, you are going to alter the distribution of electrically charged ions between the membrane, and that is going to change an aspect of the cell called its membrane potential. You're basically changing the electrical balance between the two sides of the membrane, and that's going to have huge implications for when we start talking about the nervous system, since we understand that the nervous system is all about sending electrical messages. And generally speaking, the type of signaling molecule that will open an ion channel is going to be a neurotransmitter. So we will have a lot more to say about that when we start talking about the nervous system. Okay, so the last thing that I want to talk about in this video is a real physiological example. So something that we need to understand here that kind of dates back to that video on the relationship between a ligand and its receptor. So I gave the kind of feeling that, okay, there's generally only one ligand that goes with a receptor, and that generally is true. So if you, again, consider the lock that opens the front door of your house, there's only going to be one key for that, right? Generally speaking. But some ligands can actually have multiple receptors. So imagine that the key to your front door also opens your back door, right? That makes sense, right? They're two different locks, but that one key can open them both. So since this is a virtual online lecture, this what I'm about to say is not going to have quite the same impact, but in a face-to-face class, I would generally, at this point, pull my keys out of my pocket, and I would show the class the master key to the building that we teach in. So that key could open, say, the front door of the building. It could open my office. It could open the science and math office so I can go get supplies anytime I need them. So that one key is actually capable of opening multiple different locks. Now, that's not to say a lock can be opened by multiple different keys, but one key can actually open multiple different locks. And in this way, a signaling molecule, a ligand, can actually activate several different types of receptors. A perfect example of this is a hormone called epinephrine. You might know it better as adrenaline, but we are going to call it epinephrine. So epinephrine is a hormone that is released by the adrenal medulla, and we'll learn a lot more about that in chapter 17. So epinephrine is going to basically mediate all the effects of what we call the fight or flight response. Basically what happens when you get stressed out or you start exercising, or my favorite example, if something starts chasing you. Now, you don't get chased by something every day, but that's definitely a kind of extreme example of something that would activate your fight or flight response. So what the fight or flight response is all about is preparing your body for physical, strenuous activity, some type of stress emergency. So the general idea here that we're going to look at of what we want to accomplish in the fight or flight response is we want to change blood flow. We want to get blood flowing, understanding that blood contains all the goodies that our cells need, like oxygen and other types of nutrients. We want to get blood flowing to the cells and the tissues that need them the most. And during the fight or flight response, that's going to be the brain and especially your skeletal muscles. If you're running from something like, say, a grizzly bear's chasing you, you definitely want your muscles working at peak efficiency. So we want to get as much blood flowing there as possible. So the process of opening up a blood vessel to promote more blood flow is called vasodilation. Essentially, we open up or dilate a blood vessel to get more blood flowing into a particular tissue. Well, there's only so much blood in the body, so if you do vasodilate and get extra blood flowing into the skeletal muscle, you have to do it at the expense of blood flowing somewhere else. In this case, we are also going to vasoconstrict some blood vessels and cut off the blood flowing to organs and tissues that maybe are not as important in the fight or flight response. So some things that you don't need to be focusing on if you're running from a grizzly bear: you don't need to be focusing on digesting your lunch, so maybe we could cut off blood flow to the intestines. You don't need to be worrying about making your urine, so we could maybe cut off blood flow to the kidneys, temporarily, of course. So what you're seeing here is how a single signaling molecule, a single hormone like epinephrine, can do all of these different things simultaneously by binding to several different types of receptors. So what you're seeing here is that when epinephrine is secreted by the adrenal medulla, and once it gets into the blood, when it gets into the blood, it's getting all over the place, and it's going to go find cells and tissues that have epinephrine receptors. Well, epinephrine receptors come in two major varieties. There are alpha receptors and there are beta receptors. When epinephrine binds to tissues that have alpha receptors, the effect of that smooth muscle that you find in the blood vessels is going to be vasoconstriction. So when epinephrine binds to those alpha receptors, we cut off blood flow to places like the kidneys and the intestines. But when epinephrine binds to a completely different receptor that is also an epinephrine receptor, the effect will instead be vasodilation. So here we're talking about blood vessels, arteries that lead into the brain and into skeletal muscle. So this is a perfect example of how a single ligand can get several different things happening in the body, but in order to do that, it has to be able to bind to several different types of receptors. If a ligand only binds to one very specific receptor, that ligand can only do one very specific thing. But if a ligand can bind to 5 different types of receptors, it can potentially do 5 different things, depending on which parts of the body have which different type of receptor. All right, so that is going to do it for this particular video. It was a little lengthier, so I thank you for your attention. So join us next time, and we are going to start talking a little bit more about ligands and receptors and introduce a new term called agonists and antagonists, and we'll see how that relates to the pharmaceutical drugs that we take during our lives. So thanks again for your attention, and I will